Welcome to the PFS Network YouTube channel. My name is Mitch, and today I'm very pleased to be joined by Dr. Alfonso Urbanucci, who is the group leader within the Prostate Cancer Research Center at the Faculty of Medicine and Health Technology within Tampere University in Finland, and also leads research projects at the Oslo University Hospital in Norway. Dr. Urbanucci is currently researching prostate cancer at a single cell analysis resolution with emphasis on tumor microenvironment and radiobiology. Dr. Urbanucci is an expert on the androgen receptor and has previously been published in some of the world's leading scientific peer reviewed journals. Dr. Urbanucci was key in organizing the first PFS Network scientific meeting in Tampere in Finland, which then led to the expansion of our scientific advisory board. He will be the lead investigator on our upcoming study called Investigating Genetic Factors Involved in the Development and Onset of Post-Finasteride Syndrome, which sample collection is underway for. Alfonso, thank you for bearing with me during that very long introduction, and thank you for speaking with me.
Thank you for the very nice introduction, Mitch, and it's a pleasure to be here with you today.
Absolutely. Excited to talk about your work. So, maybe first, can you give everyone a bit of background about your career and what your work is focused on?
I started my research actually here in Tampere. I took my doctoral degree in cancer genetics and molecular biology of cancer, specializing and focusing my research on prostate cancer. Then I moved to Oslo as a postdoctoral researcher at the Norwegian Center for Molecular Medicine at that time, which is a EMBL sort of a laboratory in Europe. And then I started a project group at the Oslo University Hospital, focusing on prostate cancer research still and on various aspects of radiobiology of prostate cancer. So how the cancer sort of responds to radiation therapy, and trying to improve ways to treat the patients with radiotherapies and render more effective the radiotherapy. Then I started getting interested into single cell, and with this new technology coming in, so we focused our attention on really the trying to dissect at the single cell level this the biology of the tumor, and various genomics and genetics aspects linked to the biology of prostate cancer in that sense. And of course, there are many projects that are ongoing around this topic, but of course, one of them during the pandemic became the PFS, and that's what we're gonna hopefully talk a little bit more about today. So.
Yeah, absolutely. So that segues into my next question, and you're an expert on the androgen receptor. So, can you explain to us the role of the AR and how it might have relevance to physical, neurological, and sexual symptoms after androgen deprivation? And can you tell us what happens when the AR isn't functioning correctly?
Yeah, so I must say that I became interested in the role of the AR in disease because I studied the prostate cancer. Obviously, in prostate cancer, AR is a key target for treatment, and it usually the androgen receptor in any given cell that expresses the androgen receptor works so that it links it caps the level of androgens into the cell, and the androgens namely are testosterone and dihydrotestosterone, but testosterone is what is actually in circulation, sort of in the human body. and then the testosterone usually gets into the cell, gets converted into dihydrotestosterone by an enzyme which is called 5-alpha reductase, and then the androgen receptor is able to activate because of the presence of the testosterone or the dihydrotestosterone, and gets into the nucleus, where it activates the transcription of several genes, sort of, that then do something into the cell. So in prostate cancer, the activity of the androgen receptor is aberrant, meaning that it is altered and it stimulates the growth of cells abnormally. So that is the actual point. But the androgen receptor is not only expressed in prostate cells or in prostate cancer cells, and of course the testosterone is in circulation in every um, human male person in abundance, and so there is a high likelihood that every cell that or tissue that expresses androgen receptor at physiological level will be under the effect of testosterone or hormones in general. So any activity that aims to kind of alter this equilibrium between the testosterone and the hydrotestosterone and the androgen receptor will have obviously impact in all the tissues that express the androgen receptor at the physiological level. So that is sort of kind of the first key element possibly to understand post-Finasteride syndrome.
Yeah, so basically, the AR is expressed across all these different tissues in the body. So, therefore, if you deprive the body of those androgens, then, theoretically speaking, all of these tissues could be negatively affected. You kind of have this equilibrium where you need things to be just right for androgen expression to occur correctly,
For
basically.
androgen receptor activities to be sort of
Mm,
doing whatever they do in the tissue. So the key question also to understand here is the androgen receptor is really studied in research in prostate cancer
mm,
because, as I said, is a key drug target. It is probably now starting to be studied also in other types of cancers because the researchers have noticed that the androgen receptor is also present, for instance, in breast cancer cells,
mm.
and can be also taught as a key treatment strategy for those in lung cancer. But the androgen receptor physiologically is expressed, meaning like in normal condition, not only in cancer, is expressed, for instance, in brain, in muscles, and in other tissue throughout our organs, sort of. So when we think about like a disease which could be due to aberrant activity of androgen receptor, we could think of a systemic disease that it is actually bringing several different symptoms because of this physiological expression in different tissues. So neural symptoms and muscle related symptoms, as well as sexual dysfunctions, are all sort of linked to the fact that the androgen receptor is expressed in different tissue, and through the activation, all these functions are mediated.
Yeah, yeah. And that's probably another good segue into my next question, which is why you decided to get involved in our issue, in the issue of PFS. It's probably not obvious why someone who's an expert in prostate cancer on the surface would want to get involved in our issue. So, how does your expertise lend itself here to what's like a pretty poorly understood disease, which on the surface, like I said, probably appears completely unrelated to your previous work?
Yeah, so as I mentioned, I studied prostate. I started my doctoral studies on prostate cancer, looking at the androgen receptor uh, um, function in prostate cancer cells. And in my initial studies, at how the androgen receptor overexpression alters the way the cells responds to different level of androgens, and so what came immediately clear was that if you have a given cell and you expose the cell to a certain level of androgens, and you modulate the expression of the androgen receptor, the overall sort of growth pattern of this, in my case, the prostate cancer cell, is not really the same. So if you have more androgens in androgen receptor overexpression setting, then the cell might actually found too much androgens toxic. But if you have androgen receptor overexpression and you expose the cell to a very low concentration of androgen, the cell actually grows just fine and even better than in a normal sort of a level of androgens, and in a situation in which the androgen receptor is expressed sort of in a naive way, sort of in a normal way. So that was kind of a key experiment, and that got me started to understand a little bit more about the mechanism, the molecular mechanisms that are behind this sort of equilibrium, if you want to call it that way, that they're supposed to be within the cells and possibly within tumor cells, but also this, if you want to call it also homeostasis between the sort of androgen receptor and the androgens in every other tissue. So when the question was presented to me by the PFS network, so some studies or some evidence that suggest that the androgen receptor might be involved, the overexpression of it might be involved into the onset of this condition, I soon got triggered, because indeed, although there is really nothing known about how this dynamics actually happen in other tissues, still one can think that one rules apply to everything. So one starts making sort of hypotheses about how this could happen. We are lucky because we have a very good relationship with the PFS network, and I got interested, and because of my progressed sort of experience with this beast, which I call the androgen receptor, from a scientific standpoint, is very important to understand this key aspect of this equilibrium that shouldn't be perturbed in and what I think could be one of the causes of this onset.
Yeah.
Of course, when I got to know that this condition exists, because I wasn't aware of this, I soon kind of got intrigued of why, some is it so rare. And I thought that one of the first thing we should have done was and here probably we dig into why I'm actually leading the genetic study. So why is it that some patients actually experience this disease and some other don't? And I thought that the key could be to identify like some variants in our DNA that could explain this interaction between the drug and the specific sort of genetic makeup that could allow this specific disease onset. And it is not, of course, trivial, but it would be interesting to see if there is specific variation associated of the DNA associated with some particular pathways other than the androgen receptor, for instance, but at least to let us to point at things that uh, um, could be at the basis of new therapeutic approach. So
Mm.
this could be one way. Obviously, there are many other ways to tackle the problem here. But this is what I thought, since there is very little known about it, would be a good start to ask questions related to the disease, and it kind of gets me a little bit more far away from my field of studying this equilibrium between the androgens and androgen receptor. And I'm not an endocrinologist, but I was happy to have endocrinologists on board that potentially, after the study is done, could be able to sort of comment on the results of the study. So
Mm.
so I really look forward to be able to tell you something more when this will be completed, sort of.
Well, we're all looking forward to when we get some data from your lab. So, and you're right, it is a very rare disease. So, genetics is one of the first things that comes to mind. So, can you tell us in very basic terms what this study is going to be looking at?
Yeah, so we're collecting DNA, germline DNA from patients, and we are aiming to understand whether the patients, that especially those that have been experiencing the worst symptoms, so that we are really sort of sure that they have the disease, and of course because they've taken like some 5 alpha reductase inhibitors. And we would like to do an analysis, a genetic analysis, to understand what is it that the patients have in common in terms of DNA variants, and if they have all a specific variant that could be then linked to the onset of the disease. If, of course, they have all a specific variant or a set of variants that would kind of determine a profile of risk for taking drugs of this type, then this would actually lead us to understand that, for instance, people with this genetic makeup, they should not be exposed to the drug, because in those patients the risk associated to taking the drug are very high, including the development of PFS.
Okay, that makes sense. And Um, I think like one question that some people have about looking at genetics, or some patients have about looking at genetics, is um, they don't really necessarily care like why they were affected. They just want to know what can be done to fix this, um, or kind of like what has happened and how to fix it. Why is it important to not only focus on what's happening, but why it's happening when we think about like progression towards a possible treatment?
Yeah, absolutely. I mean, the variation of DNA that we're looking at can can be concerning genes, and these genes ultimately could be targets for treatment. So, understanding why something happens can allow us to understand how to correct the this the onset of the disease. If it is like a gene which is functioning in a way that we can actually um, reconstruct the function, or that is kind of impacted by the drug, or by the variant, then we could potentially improve the symptoms.
Hmm.
So the hope is that understanding why led us to also understand how we can how we can generate some therapeutic approach.
Yeah, because I think that's not always obvious to patients who aren't familiar with some of the scientific concepts involved, or how this kind of research can progress. Is that they just kind of think of it as like, well, I want to know like how to treat the thing, not necessarily like why it's happened. But the two, what we're doing at Kiel and what we're doing with you, they kind of go hand in hand. Like they're two different paths kind of going towards the same thing, and you need both of them happening in parallel, basically. Um,
Absolutely, the studies are connected, and we hope to get genetic information also from the study that Nadine is performing in Kiel, and with her functional studies, we would also be able to understand the impact of what is a genetic variant actually doing, and
mm,
uh, and therefore strengthen our hypothesis on whether some particular variant might lead into identification of targets that could be used for treatment.
yeah. So the study is underway. We're as of last week we've started the recruitment process or the collection process. So very excited to start getting some data over to your lab. Um, so just very quickly for people who aren't familiar, what is a whole genome sequencing?
Yeah, so every cell of our body actually has the same DNA. And so when we talk about germline DNA, we're talking about what is our genetic makeup. So the sequencing our DNA can be achieved in several ways. You can sequence the whole DNA, the whole genome, which is 3 billion bases at the several at quite a kind of unique depth that allows you to call the variants of our uh, DNA, because not every human being has the same 3 billion bases in the same sequences. So there are variation of this spots of some of the spots in the DNA, and so whole genome sequencing allows to determine like this little bits and pieces of variation of this 3 billion basis sequence, and to call the variants with quite a certainty, so that we can determine why individuals are different. So yeah.
Yeah. So you can look at the whole genome and start to pick up different variants in individuals that we're studying. Okay. So given what was found in the Baylor study, which was that the androgen receptor in I think it was 26 different PFS patients was um, overexpressed, and there was dysregulation of gene expression. Why does this study make sense as a logical next step?
Yeah, so the Baylor College study pointed at upregulation of androgen receptor in the PFS patients, and but that actually was limited to one tissue, if you want to call it that way, that was examined. So, and the reason why the upregulation of the androgen receptor is happening, it's actually not understood. So, having a genetic studies which could potentially uh, led us to understand whether there are variants in the bits of the DNA that regulate the androgen receptor and many other genes involved potentially in the signaling of androgens. It's important because we would also try to understand mechanistically what is it that causes this upregulation, if this upregulation can be actually studied in the cells in a certain way, and in a way also prioritizing the variants that are associated with upregulation of certain genes that also were found in the Baylor College study would be sort of part of this integrative analysis that we would do to to better examine like what are the genes that are involved. Yeah.
Hmm. Yeah. Okay. And one topic I wanted to discuss with you, and we've spoken about this privately before, but very curious to get your thoughts on here. Um, there's a key feature of PFS, which is kind of colloquially known as the crash, and it's basically where patients they stop taking the drug, um, they experience an improvement in on drug symptoms for a very short period of time, and then they get drastically worse. So their symptoms become far worse than when they were on the drug, or they experience quite often many new symptoms that they didn't even have while they were taking the drug. That was my personal experience. I only experienced some very mild symptoms while on the drug, stopped, improved, and then got 30 or 40 new symptoms literally overnight. Um, and that onset of this persistent disease has been experienced by about 80% of a group of 427 patients that we surveyed. So I know that's a very long introduction to the question, but in your opinion, what might be happening there?
Yeah, so I am not an endocrinologist, and I am sure that an endocrinologist would have much more medical explanation for that.
Right,
But
right.
based on what I said at the beginning of this interview, that there is an equilibrium between the androgen receptor and the androgens level, and what the drug does is to sort of disrupt this equilibrium drastically while the person is in this drug and in several tissues. Then what I think is happening during the crash is that the relief of symptoms might be due to the adjustment that the androgen receptor sort of level have in these particular tissues that are interested by the shutting down of the signaling. Uh, and so obviously many people experience different symptoms and different crashes, different symptoms within the crash because their level of androgen receptor reacts in different ways in different tissues, also according to their genetic makeup. And it is really hard to tell like why this is happening. I hope that gives an idea of why this crash might happen. So several different issues involved anyway.
Yeah, no, I think it's really interesting because it's a very it's one of the kind of key peculiarities of the disease is that you have this period where people actually feel better when they stop taking the drug in many cases, and then they completely shut down. So before we wrap up, as we move forward and we continue research into PFS, how could possible findings from your study be practically applicable to the problem, and what are some potential outcomes of this first study?
Yeah, so as I mentioned earlier, I hope that this genetic study will be able to tell us, first of all, what are the people at risk and that should not take the drug. So it would be theoretically possible to define genetic risk profile that could be associated with taking the drug, and so that's sort of preventive of other people to develop this disease. Secondly, also we hope to find variations in the DNA that would explain in certain tissues certain symptoms, and to be able to tackle those therapeutically. and so that's very important for existing patients. And of course, integrating our data with other data sets that are being produced or that have been produced already, or that are actually hidden in databases that we're digging, we can potentially stimulate more research, more hypothesis-driven research, and try to get more scientists involved into this initiative. Yeah.
Mm-hmm. Yeah, I think that's something that's really important that Nadine spoke about on our interview as well, is that there's such a small amount of data about this issue currently that really what we're trying to do here is generate some insights with these first studies to kind of be able to more appropriately form some hypotheses around what might be going on and what to look at next, rather than kind of starting at the hypothesis and working backwards. It's like looking at the whole picture, looking at all the symptoms, and then kind of figuring out what we do next. So
Yeah. What I didn't mention, and I can add here, is that one important thing is to be able to start clinical trials too.
mhm,
So if we can have a better hypothesis of what would be kind of a ideal treatment, we could test that.
mhm,
And of course, we wouldn't be going testing it without having, of course, all the regulations in place and everything, and all the permissions and so on. But this is just to say that these basic studies are also the basis of building an hypothesis that then can be tested within a lifetime, hopefully, for PFS patients today. Yeah.
yeah, absolutely. Well, Alfonso, thank you very much for speaking with me. I really appreciate your time. I know you're very busy, so thanks for taking the time to speak with us, and thank you for being involved in all of our work.
Thank you for having me, and I'll see you soon. And I hope that the PFS networks that are watching this video have enjoyed it.